welcome to today's live broadcast, RNAi success in difficult-to-transfect cells by self-delivering siRNA. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Dharmacon. Dharmacon, part of GE Healthcare, continues to be the industry leader in the field of RNAi chemistry, RNAi biology, and high-throughput screening, and partners with the RNAi screening community through participation in the RNAi Global Initiative. With Dharmacon, you get access to the largest and most complete portfolio of innovative and technologically advanced tools for gene silencing, gene expression, and gene editing. For more information, please visit www.gelifesciences.com/Dharmacon. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. I would now like to introduce today's speakers, Louise Baskin and Michael Freeley. Louise Baskin is a Senior Product Manager at Dharmacon, having joined the company in 2005. She holds responsibility for all synthetic RNA products, including custom and pre-designed siRNA and microRNA reagents. Michael Freeley is a Senior Research Fellow in the Department of Clinical Medicine in Trinity College, Dublin, Ireland. His research focuses on the cell biology of the T-cell immune response, particularly the signaling pathways and proteins that regulate T-cell activation and migration. I will now turn it over to our first speaker, Louise Baskin, for her presentation.
Thank you, Brenda, for the introduction, and thank you all for joining us today. Our topic for the next hour or so will be RNAi success in difficult-to-transfect cells by self-delivering siRNA. Before we start, we have a very quick polling question for the group. We would like to know what type of nucleic acid delivery techniques that you've used before. So please click all that apply to you. This just kind of helps give us an example of who we've attracted today and what kinds of things you might learn. I'll leave it open just for a couple more minutes while everyone gets a chance. Again, please do click all that apply to you. I'll go ahead and close it, even though we're still getting some, just so we can get moving. And I'll go ahead and show those results for everyone curious. Oops. And it's not, there we go, sorry. It's not letting me show you the results. So I guess we'll just have to look at that later. Anyway, so for our agenda today, I'm going to be starting with a very brief overview of how Dharmacon addressed issues with delivery of siRNA with our development of Accell siRNA, which is really going to be the main focus of today's talk. As part of my section, I will very briefly introduce a few notable successes that have been carried out with Accell applications. And then I'll turn it over to Michael Freeley, who will present some really great work that he and his lab have carried out in primary human T cells. And he'll give a more in-depth look into the details on how they applied Accell siRNA in a screening environment for looking at T-cell migration. Since the advent of RNA interference, or RNAi, Dharmacon's driving passion and focus has really been around excellence in RNAi tool development. One of our earliest achievements was in publishing and commercializing the first set of rational design rules for functional siRNA. And this algorithm forms the basis of all of our siRNA products. We also heavily utilized the 2' ace RNA synthesis chemistry that Dharmacon was founded upon to develop chemical modification patterns for siRNA that address the growing needs of scientists carrying out RNAi. One of our first modification patterns was SI stable, which improves siRNA stability by preventing siRNA degradation by nucleases, which is particularly useful when using siRNA in vivo. Another key development was our patented on-target plus modification pattern, which improves siRNA targeting specificity, thereby reducing off-targets. So today we're going to focus on a product that was launched a few years after on-target plus that utilizes aspects of all of these innovations, and it's called Accell siRNA. So even with the advancement in siRNA potency and specificity that came with the growth of RNAi as a mainstream tool in molecular biology, a key issue remained, and that is, how can we carry out siRNA-mediated gene silencing in cells that are resistant to standard transfection methods? So for years, we at Dharmacon had heard from customers who wanted to work in biologically relevant cell types, things like beta islet cells, cardiomyocytes, primary neurons, and T cells. We heard these requests all the time, but delivery of the siRNA was a huge challenge. Most siRNA experiments are carried out in transfectable cells, where standard cationic lipid transfection reagents are perfectly suitable. However, many cell types are highly resistant to or have a cytotoxic response to transfection reagents. Electroporation or nucleofection and other instrument-based methods are very useful for a wide range of cell types, but they can cause rather extensive cell death, and the consumables required, like buffers and cuvettes, the somewhat high amounts of siRNA, and not to mention the instrument itself, can be rather expensive. Lentiviral transduction, which delivers shRNA rather than siRNA, is a good option for non-transfectable cells. Plus, it gives you the advantage of creation of stable cell lines. However, some cell types have undesirable responses to viral transduction, and not all labs may have access to the required BSL-2 facilities. So the goal in mind when developing Accell siRNA was to eliminate the limitations of these delivery methods and instead use our RNA chemistry and cell biology expertise to create a chemical modification pattern on the siRNA itself that enables passive delivery, and particularly enable it to difficult-to-transfect cell types. Now, I'm not going to relive the entire product development cycle. That would just take much too long. Instead, I will just summarize the overall approach. So first, we wanted to ensure that the siRNA was functional. So in addition to using our smart selection algorithm for designing potent siRNA, we included a modification which prevents the sense strand of the siRNA from loading into RISC, thereby increasing the antisense or the targeting strand activity. Second, since these siRNAs are not being encapsulated in a transfection reagent, we incorporated modified bases, very much like our SI stable modification, to prevent degradation from nucleases. And lastly, and really most importantly, we had to find a way to get these siRNAs into cells without the requirement of a carrier. We settled on cholesterol, which, as you likely know, is a common component of the lipid bilayer, and it's long been used to introduce oligonucleotides into cells. Now, what was interesting to us were the significant differences in uptake we achieved depending on the type of linker used to conjugate the cholesterol to the RNA. So the aliphatic linker that we settled on gave us dramatically better delivery than all of the others that we tested. So if you're curious about any further details on the RNA chemistry itself, please feel free to look up our patents that cover this technology. In the interest of time, I'll now move on to the application of Accell siRNA. As I mentioned, a big part of our product development was ensuring that all of these modifications for delivery and stability did not interfere with effective gene knockdown or cause unwanted cytotoxicity. Now, as you can see from our early pilot data in these 7 cell lines across a few dozen different gene targets, we were able to achieve very good target silencing, which is shown by the blue bars showing the level of knockdown, and we maintained very high cell viability, shown by the red dots. Now let's talk about how to use Accell siRNA. So the Accell delivery protocol can be broken down into 2 primary steps, but please keep in mind that thorough optimization is required, just like we would recommend for any new application. So first, the Accell siRNA is diluted in low serum or ideally serum-free media to create what we call the Accell delivery mix. Now, what can sometimes confuse our customers is that we do sell a serum-free media called Accell delivery media, but they often misunderstand and think that it contains some special ingredient that is required for use, and that is not the case. Nearly any serum-free media or growth media with its serum content adjusted should be fine. It's really the Accell siRNA itself that enables the delivery. Now, as for working concentration, typical siRNA experiments can be run with anywhere from 5 to 50 nanomolar of siRNA. For Accell, we tend to see best results with 200 nanomolar up to 1 micromolar of siRNA. Now, while that might seem alarmingly high to those of you accustomed to using transfection reagents in HeLa cells, for example, remember that this is a totally different molecule using passive delivery, which is generally less efficient than other methods. So the amount that actually gets into the cell when working at 1 micromolar is very similar to what you would see with conventional siRNA being delivered by transfection reagents at a lower concentration. Now, once you've worked through the appropriate conditions for the delivery mix, you simply add it to your cells. Now, here's where we see another deviation from standard siRNA transfection. We do recommend a longer time point prior to assaying for knockdown. For instance, with standard transfection, we can typically see mRNA knockdown by 24 to 48 hours. But with Accell, we push that out to 72 to 96 hours, and even longer for protein detection. Again, this is a difference due to that passive delivery. The overall duration of silencing will still be the same, around 4 to 6 days. And you can add serum-containing media back to your cells after about 48 hours if they're showing some sensitivity. So now that I've given you a very high-level idea of how to use Accell siRNA, I want to show you a few examples of Accell in action. Now, the examples I chose are all neuronal cell models, but please do keep in mind that many different cell types have been successfully used with Accell siRNA. So this first example is an experiment that was carried out in the early days of Accell as a proof of principle. Primary neurons were one of the cell types that we heard about a lot from customers in terms of the limitations they were running into with use of siRNA. So we worked with a group that was wanting to use primary neurons as a cellular model for Alzheimer's disease. However, again, delivery of siRNA to these primary neurons was really an obstacle for them. So this slide provides an overview of the protocol that was used. So on the upper left, you'll see the types of Accell labeled and non-labeled controls that were used, as well as an Accell Smart Pool reagent targeting P53, which is being assessed as a positive control. And these were combined with neurobasal media with a B27 supplement, and then were then added to rat cortical neurons. So this use of neurobasal media is a perfect example of a serum-free media that would be used for growing these cells anyway. So the addition of Accell siRNA to the experiment was really not very disruptive. They then incubated the neurons for 2 different time points and then assessed for uptake of the Accell siRNA by visual inspection of the labeled controls. And they also assayed for target gene mRNA knockdown. So first we'll look at how the labeled Accell red siRNA controlled localized in the treated cells. As you can see in panels A and B, both the wide and the, A and C, the wide and the detailed views, virtually all of the neurons were positive for uptake of the Accell red control, and that uptake was really throughout the cell body and out into the neurites. Next, they looked at p53 knockdown on the mRNA level. As you can see, at the 48-hour time point, there was a reduction of p53 by over 60%. Now, this is a somewhat early time point based on what we would now recommend for Accell application. It would be expected the knockdown would be even greater at a later time point. Now, while there was still a fair amount of optimization to be done, these researchers were able to conclude that Accell would permit them to carry out these functional target validation studies in primary cortical neurons. And this was a cell type that had been unusable in this manner up until this point. Next up is a very similar protocol, but this time the researchers were working in organotypic brain slices. So this is a model that permits investigators to maintain all of the cell types within a heterogeneous tissue, and it more closely models the in vivo cellular circuitry, but in a more convenient ex vivo environment. So in this study, again, an early proof of principle for this interesting but difficult to transfect tissue model, our collaborators incubated mouse brain slices in Accell controls that were diluted in, this time they used Accell delivery media, but they did add the B27 neuronal supplement. They then carried out both visual analysis to verify uptake of the labeled control, as well as Western blot analysis to look at protein level of the target gene. So here are photos from 8 hours and 72 hours post-application of the labeled Accell siRNA. And you can see after 72 hours in culture, fluorescence in the slices was much more intense. It really indicated an increased uptake of the Accell siRNA into the deeper layers of the tissue with that extended incubation. The Western blot analysis was similarly encouraging. As you can see in the upper right, glycophilin B showed significant depletion at the protein level by the 96-hour time point. So as with the primary neurons, this was a very encouraging demonstration of Accell siRNA, enabling gene knockdown in this difficult-to-transfect tissue model. My last example is from a much more recent publication, and I think really highlights this culmination of successes in the other neuronal systems that came before. So this group from the University of Guelph in Ontario carried out a study of DNA methyltransferases and their impact on object memory processing. And they did this by selectively knocking down 3 different DNMT family members with Accell siRNA in different regions of the brain, and then assessing their subjects to object-in-place learning. The siRNA was delivered via a cannula to either the hippocampus or the perirhinal cortex. Now, I cannot possibly do justice to the entire breadth of this paper. I would encourage you to reference the full article for details. I do just want to point out 2 data points that highlight the Accell siRNA results, specifically the targeted mRNA knockdown and how they were able to correlate that knockdown to the behavioral assessment of the subjects. So this first figure shows the mRNA knockdown levels at 48 hours after a single dose of Accell siRNA to the CA region of the hippocampus. So the CA region was isolated, and the mRNA levels for each of the indicated DNMT targets was assessed. So as you can see, there was a very good discrimination between the 3 different family members and very good knockdown levels overall. And now here's one of many examples from the paper of how targeted knockdown impacted the behavioral phenotype. So in subjects that were treated with Accell siRNA targeting, in this case, DNMT3A, there was virtually no difference in their long-term memory formation immediately following siRNA administration. And so that's what's shown in the bars on the left. But 24 hours after administration, as you can see from the bars on the right, show that there was significant impairment observed in the subjects that were treated with Accell targeting DNMT3A compared to the control. Very similar results were also seen at the 48-hour time point, demonstrating to this group that Accell siRNAs were active in vivo both 1 and 2 days following the administration. There have been many other published successes with Accell siRNA in vivo, and we're very happy to continue to see these results where it's used for successful in vivo target validation. So that brings me to the end of my portion of today's webinar. I'll turn it over to Michael in just a moment. Hopefully you've gained an initial understanding of Accell siRNA, how it's delivered to cells by way of chemical modifications rather than transfection reagents. For anyone interested in giving Accell a try, it is available pre-designed for human, mouse, and rat. You simply search for your gene on our website. If you're working in a different species, we can apply Accell modifications by way of custom synthesis. So very easy to get this for nearly any species you might be working in. So without any further delay, I want to get to what I consider to be the most interesting part of the webinar today. Michael Freeley is going to discuss a screen that his team carried out in primary T cells using Accell siRNA.
Okay, thank you very much, Louise. So the title of my talk today is RNAi screening for identification of genes that regulate primary human T cell migration. So T cells play a very important role in defense against pathogens and tumors. And one of the cornerstones of immunology and, of course, of T cells is that they can discriminate between non-self, such as the pathogen and tumor, and self tissues. However, in some patients, this discrimination between self and non-self can be lost. And this can lead to inflammatory disease or autoimmune disease. As well as that, unregulated T cell proliferation can lead to many T cell leukemias and T cell lymphomas. Whereas last but not least, the T cell is the site of infection by the HIV virus, and this is a global problem still to date. So really, understanding the genes and signaling pathways that regulate T cell function is very important from both a basic biological viewpoint and also from a therapeutic standing. So one of the ways we can understand the function of genes in T cells is by a silencing of gene expression approach using siRNAs. Now, primary T cells are known as hard to transfect, and that's because they're suspension cells and they're not very transfectable with lipid-mediated transfection reagents. So that creates a problem. So two of the most common methods of delivery of siRNAs into T cells is electroporation and nucleofection, or viral vectors. Electroporation creates transient pores in the plasma membrane of the T cell, and this allows the siRNAs to traverse into the cytoplasm and cause the silencing of gene expression. However, a notable side effect of this electroporation is the loss of cell viability. The second most common method of delivery are viral vectors, and this is where the siRNA is usually encoded in the genome of the virus as a short hairpin RNA. And subsequent incubation of the virus with the T cell allows the shRNA to be expressed in the T cell. Some less common methods of siRNA delivery to primary T cells are conjugation of the siRNA to a peptide or protein, to nanoparticles, or to aptamers, which enable efficient delivery of the siRNA into T cells. And last but not least, as Louise introduced, the new player in the market are Dharmacon Accell siRNAs, and these are chemically modified siRNAs that enter into cells without the need for a transfection reagent. And for those of you who are interested, we've recently reviewed the methods of siRNA delivery into T cells for both applications in inflammatory disease, cancer, and infection. So with the discovery of RNAi and the development of RNAi libraries, this really transformed the field as it enabled rapid and unbiased novel gene discovery application processes to come to the fore. And this busy slide summarizes all of the siRNA screens that have been done in T cells to date in vitro. And many of these assays, they focused on genes regulating NF-kappa B activation, for example, genes regulating T cell migration, and also genes regulating T cell proliferation and survival. However, what you'll notice from all of these screens done to date was that they all used either nucleofection and electroporation or lentiviral vectors to carry out these screens. And this creates a major problem because, of course, carrying out an RNAi screen is such a big way requires scale-up. And this is not very easy when using reagents like nucleofection or lentiviral vectors. For example, the use of nucleofection for RNAi screening requires multi-well electroporators. And of course, as I mentioned earlier, you can get loss of cell viability with this. As well as that, using lentiviral vectors requires expertise, knowledge, and biosafety levels to carry out these screens. And again, this is not very easy. Another point I'd like to make is that a lot of these screens used transformed T-cell lines, such as the JERCA T-cell, to carry out these screens. And it's well known that JERCA T-cells harbor many mutations in their signaling pathways, and thereby they're not reflective of primary human T-cells. So really, a major advance in the field would be to be able to perform RNAi screens easily in primary human T-cells using much easier methods of delivery other than nucleofection or lentiviral vectors. So our research is focused on the molecular mechanism of T-cell migration and activation. So for T-cells to become activated, they must migrate from the bloodstream into secondary lymphoid organs, such as the spleen or lymph node, where they meet antigen-presenting cells. And the first step in this cascade is the arrest of the T-cell on the blood vessel wall. This arrest is mediated by chemokines, which are immobilized on the surface of endothelial cells near the entry point to the lymph node or spleen. And these chemokines signal through their cognate chemokine receptor expressed on T-cells. And this induces a rapid intracellular signaling cascade that converts adhesion receptors, such as the integrin LFA1, from a low affinity to a high affinity conformation. And this conformational change in LFA1 permits the T-cell to bind to its counterligand ICAM1, which is also expressed on the surface of the endothelial cell. And it's this cascade that enables T-cell arrest on the blood vessel wall. As well as that, integrins such as LFA1 can signal bidirectionally, such that the ligand-bound form of LFA1 can signal back into the T-cell, and this can cause polymerization and redistribution of the cytoskeleton in T-cells. And this results in a functional change in the shape of T-cells, and they change shape from a round spherical shape to a tadpole-like morphology, and they start actively migrating along the blood vessel wall until such time as that they meet a suitable entry site into the secondary lymphoid organ. Once within the lymphoid organ, they follow chemokine gradients until they meet the antigen-presenting cell, which will be presenting on its surface part of the pathogen. Over the course of a number of days, that T-cell may become activated and differentiate into what is known as an effector T-cell. And these T-cells have new properties. For example, effector CD4 T-cells acquire the ability to secrete distinct inflammatory cytokines, whereas effector CD8 T-cells now acquire the property to directly kill target cells, infected target cells and tissues. As well as that, the T-cell, once it's in the lymph node, it must be able to migrate from that lymph node back into the bloodstream and into the peripheral tissues where the pathogen was first encountered. So a similar mechanism for migration into these peripheral tissues, such as the skin and gastrointestinal tract, is also mediated by chemokines and integrins, such as LFA1. So in our lab, we have a model of T-cell migration, and that's whereby we take primary human CD3 T-cells and we activate them over the course of a number of days with phytohemagglutinin and the T-cell growth factor interleukin-2. And this pre-activation mimics T-cell activation in lymph nodes by antigen-presenting cells, and thus we generate effector T-cell populations. We then take the cells and we incubate them on recombinant ICAM, usually in the presence of an immobilized chemokine, and this stimulates the T-cell polarization that I talked about previously. As well as that, we have another model whereby we take a cross-linking anti-LFA1 antibody and immobilize it on the surface of a plate, and placing the effector T-cells on this substrate also induces T-cell polarization. So we can make videos of this T-cell polarization, and now I'm going to show you a video of what actually happens when we track the cells using the in-cell analyzer live cell microscopy. So I hope everybody was able to see that T-cell migration video. You can see the T-cells actively migrating over the substrate of the plate. So we were interested in trying to identify novel genes regulating T-cell migration. So with that in mind, we wanted to perform some RNAi screening of some libraries to identify genes that regulate this process. And once we started out in this field, we came into the usual problems of the RNAi delivery into primary human T cells. For example, we had to decide would we use electroporation or would we use viral vectors to deliver the RNAi library into T cells. And luckily about that time, Dharmacon released their Accell RNAi technology, and of course these Accell siRNAs are available in library format. So we wanted to use an Accell siRNA library as a proof of principle strategy to identify genes involved in LFA1-mediated T cell migration in primary human T cells. And the work I'm going to describe to you today was just recently accepted in the Journal of Biomolecular Screening. We received an Accell siRNA library from Dharmacon, and this library was a small siRNA library targeting 72 distinct genes. Now these siRNAs in the plate were SmartPool siRNAs, so they consisted of 4 distinct siRNAs targeting each distinct messenger RNA in the cells. And the library was configured to target a variety of intracellular signaling pathways. And this library plate map in light blue, for example, shows the kinases which were targeted, shown here. As well as that on the plate, there was an siRNA targeting the dehydrogenase GAPDH. There was an siRNA targeting the small GTPase ROA. And importantly, when doing library screening, you also have to have negative controls. So we had a non-targeting siRNA pool in Accell format, as well as the 4 individual siRNAs non-targeting pools from this smart pool. In addition, we also had a fluorescently labeled or FAM-labeled siRNA targeting PPIB, and this reagent was used as a useful reagent to monitor siRNA delivery into the primary human T cells. So after we received the Accell library from Dharmacon, it was received in a dried-down format, 1 nanomolar Accell siRNA per well. We added 100 microliters of the Dharmacon siRNA buffer to each well of the plate, to the master plate. And this gave a concentration of 10 micromolar siRNA per well. Then using a handheld pipette, we produced 10 daughter plates from this library, each with 10 microliters of the Accell siRNA per well, and these were stored at -20 until the library screening procedure. As Louise described earlier, adding the Accell siRNAs to your cells is quite easy. You just resuspend your cells at the required concentration in delivery medium and just add the Accell siRNA. After incubating the cells for a required period of time, you can measure things like delivery of the Accell siRNAs by flow cytometry or confocal. You can analyze knockdown of a target gene of interest by qPCR or Western blot, and or you can measure a cellular phenotype. So we did something like this, but because our siRNAs were already on the plate, we actually just added our T cells in Accell delivery medium directly to the siRNA daughter library plate. And these cells were resuspended at the recommended concentration by Dharmacon of 2 million cells per mil. And after incubating the cells with the library for 72 hours at 37 degrees, we did all of these assays. We looked at delivery of the Accell siRNAs by flow cytometry and confocal microscopy. We monitored knockdown of target genes by qPCR and Western blot. And we also importantly looked at a cellular phenotype, in our case, the T cell polarization on anti-LFA1, using an image-based high-content analysis platform. So the first part of the work we did after we transfected the cells with the Accell siRNA library was to confirm delivery of the siRNAs into the cells. And as I mentioned earlier, one of the siRNAs in the library was a FAM-labeled siRNA targeting the PPIB. So we harvested the cells from this plate and we washed them and analyzed them by flow cytometry. And you can see here on this histogram plot on the left that 100% of the primary human T cells were transfected with the FAM-labeled PPIB siRNA. Whereas on the right, this is a negative control, which is a non-labeled siRNA targeting PPIB, just to show the background fluorescent levels. This showed that the siRNAs were taking up into the T cells. When we submitted this work, we actually had a question from a reviewer who wanted to know were the siRNAs just stuck to the outside of the cells. And of course, this could lead to a false positive result by flow cytometry. So to address this question, we also looked at Accell green siRNAs, which, like the FAM-labeled PPIB, can be used for subcellular localization studies. So we treated transfected T cells with the Accell green siRNAs, and this time we fixed them and stained them with both a antibody to the LFA1 integrin in red here. And you can see here the Accell green siRNA by confocal microscopy and looking at Z-stacks of the cells are clearly delivered into the cytoplasm of primary human T cells. Whereas on the right here shows a negative control experiment whereby T cells are incubated with a non-labeled control siRNA, which has no fluorescence, just provides background reference control. And of course, these siRNAs were uptaken into primary human T cells as well by flow cytometry, Accell green siRNA. So that was the first question we addressed. The second question was to verify knockdown of a target gene by either qPCR or Western blotting. And as I mentioned earlier, one of the siRNAs in the library targeted the GAPDH molecule. So we harvested these cells, and after 72 hours we could see that the siRNA targeting GAPDH resulted in approximately 70% knockdown of that target gene at the messenger RNA level in comparison to control experiment setup that were treated with an Accell non-targeting control siRNA or T cells non-treated with any siRNA at all. Again, this is a very positive result. The last part of the study we looked at in this screening assay was to look at a cellular phenotype, and in our case the change in cell shape from a round shape to this polarized T cell morphology. And we used an image-based high-content analysis to monitor this screening. So after 72 hours, approximately 20% of the cells from the library were taken out from the library plate and incubated on an immobilized anti-LFA1 antibody. And these cells were allowed to develop a polarized morphology, similar to the video that you hopefully saw earlier. The cells were then fixed with paraformaldehyde and stained with a Huxt stain to label the nucleus, an antibody targeting tubulin linked to FITC fluorescent molecule in green to label the alpha tubulin cytoskeleton, and also the cells were labeled with phalloidin linked to TRITC to label filamentous actin. And here you can see a representative image from the in-cell analyzer showing the polarized T cells. And of course, to quantify this T cell morphology, we didn't want to do this by hand, so we used an in-cell analyzer from GE Healthcare to both acquire and analyze these images. And this was very important because this screening and image analysis procedure allowed us to quantify roughly 8 to 10,000 cells per well, which is very important when you're doing knockdown experiments to show that you need to cover a cell population in your analysis and not just cherry pick some cells. So the in-cell analyzer allowed us to automatically acquire all of these images, and it also allowed us to analyze the cell morphology in an automated manner as well, which I'll explain in the next slide. So for those of you who are not familiar with the in-cell analyzer, it detects cells by nuclear dye uptake, shown here, and it measures the morphology of cells based on a number of cellular stains that you can use. So in our case, we had stained the cells with an antibody to alpha tubulin and the phalloidin TRITC to label the filamentous actin. So we based the T cell morphology analysis on both of these stains. And of course, there's many ways to measure T cell morphology, but for T cells we routinely use 4 morphology parameters, which we have previously shown in a publication to be most informative for analyzing T cell shape. And I'll go through some of these. The most important one that we find is the 1/4 form factor parameter, which is a 1/4 roundness index. And we find that as T cells polarize and migrate from a round spherical shape to a polarized elongated morphology, that the 1/4 form factor parameter increases. And it's a good readout of T cell migration. The second parameter that we use is nuclear displacement, and this measures the position of the nucleus relative to the center of the cell body. So in non-migrating or non-polarized T cells, the nucleus is usually found localized in the center of the cell, so it's not displaced from the cell body. However, in T cells that are actively migrating, the nucleus migrates and it moves to the front of the cell and away from the center of the cell body, and thereby nuclear displacement also increases. We also use the generic morphology parameter cell area, which also increases during T cell migration, and the cell gyration radius, which measures the spread of a cell. And all of these parameters increased as well. So there are good readouts of T cell migration. The in-cell analyzer also comes with a number of fluorescent intensity parameters, which allowed us to measure the fluorescent intensity of either the alpha-tubulin or the filamentous actin stain, and this was mediated by the IXA N+C parameter. As well as that, we also included some other informative parameters such as intensity spreading, which allows you to measure the amount of staining near the cell boundary, as well as the cell intensity CV parameter, which allows you to check how homogeneous your staining is inside the cell. And these, again, were useful readouts of T-cell morphology. So we performed 3 RNAi screens using T-cells from independent donors, and we identified siRNAs that affected the various T-cell morphology parameters using a Z-score system. So for those of you who are not familiar with a Z-score, it's a common statistical method for analysis of high-throughput screens, and it expresses a sample value as the distance from the mean in multiples of standard deviations. So we defined the effects of siRNAs as being statistically significant only if they exceeded a Z-score threshold of +2 or -2 in 3 out of the 3 screens. And we displayed the Z-score data as a heat map shown here on the left. And the Z-score data was, once it was displayed as a heat map, they were coloured. So siRNAs with a Z-score of -2 or greater were coloured in green, while siRNAs with a Z-score of +2 or greater were coloured in red. And from the 3 screens performed, you can see here quite clearly that the siRNA targeting the small GTPase RhoA, it increased the Z-scores for T-cells migrating on anti-LFA1, whereas because the 1/4 form factor and the nuclear displacement parameter was increased for both the tubulin and the filamentous actin stain. So this indicated to us on the screen that once we treated the cells with the siRNA targeting RhoA, that these cells elongated relative to all other samples on the plate. And indeed, visual inspection of the RhoA treated cells shown here show that these cells had elongated tails relative to the control siRNAs, which is a nice result. As well as that, our FAM-labeled PPIB siRNA, although not affecting any of the T-cell morphology parameters, it did increase the fluorescent intensity parameters, particularly only of the alpha-tubulin stain. And this makes sense because the alpha-tubulin stain is in green, and therefore when these cells are also treated with a FAM-labeled PPIB, which is also in green, this increases the fluorescent intensity. So this confirmed the siRNA delivery to the primary human T-cells and complements the studies we showed using flow cytometry and confocal microscopy. So after performing the 3 screens, RhoA was one of the hits from that library. However, you may notice that we only used 20% of the cells from the library for the migration assay. And of course, an advantage of using suspension cells, such as primary T cells in RNAi screening applications, is that the remainder of cells can be used for additional assays without requiring extensive manipulation, such as trypsinization. So because we had quite a few cells left over, we used the remainder of cells for 4 additional assays. And these assays were cell viability, whereby we added cell titer blue reagent to 20% of the cells. We also measured total cell count using a high content analysis procedure, again staining the cells with the nuclear dye Hux 33258. And we also measured adhesion of the cells following knockdown to ICAM1, either in the absence or presence of the chemokine SDF1. And this is important because adhesion of T cells is the step that occurs prior to migration. So we also wanted to look if any of the genes affected this process. So this slide summarizes the siRNA screens using the cell titer blue, cell titer total cell count, and adhesion assays that we did. And again, we used the Z-score threshold to define hits. So we're looking for anything that's +2 or -2 as a hit. However, we did not find any of the siRNAs affected any of these parameters in these assays. So there was no hits from these particular screens. So going back to our migration assay, we identified siRNAs targeting RhoA, and we found that they affected the T-cell migration. So with that, we had to validate this work. So using T-cells from independent donors, we transfected them with a control siRNA or an siRNA smart pool targeting RhoA. And after 72 hours, the cells were harvested and lysed and then probed by Western blotting with an antibody specific for RhoA. And you should be able to see there on the top panel that RhoA was knocked down by the RhoA smart pool after 72 hours. As well as that, we also looked at the 4 individual siRNAs from that RNAi SMART pool to test whether if each or all of these had an effect on the RNAi expression levels. And we found that 3 out of the 4 siRNAs shown here resulted in knockdown of RNAi expression. RNAi siRNA number 4 was the least effective in our hands. And quantification of the RNAi knockdowns from 5 independent donors showed that the siRNAs targeting RNAi resulted in approximately 80 to 90% knockdown of RNAi at the Western blot level in our hands. Of course, once we knocked down RNAi, we had to show that these cells would elongate more relative to controls. So again, the same cells from these Western blot experiments were plated on an anti-LFA1 antibody, shown in the black bars here, or as a negative control, they were plated on poly-L-lysine, which induces T-cell adhesion but does not induce the migration phenotype. And I hope I can show you there that the RNAi siRNA, treating T-cells with the RNAi siRNA induced an elongated morphology using the 1/4 form factor parameter relative to the control siRNA treated cells or non-transfected cells. As well as that, the 3 out of the 4 siRNAs here targeting RNAi also produced a similar phenotype of increased T-cell polarization. And a very identical result was also seen when nuclear displacement parameter was chosen as the morphology readout instead of 1/4 form factor. So knocking down RNAi induces an elongated phenotype. And this next slide shows some of the images from the in-cell analyzer. So these cells again had been stained with phalloidin TRITC and alpha-tubulin green. And hopefully you can see in these images that the elongated tails of the T cells once RNAi expression had been suppressed. And this was evident through both the SMART pool targeting RNAi and the individual siRNAs as well. This next slide shows the level of RNAi silencing correlates with cellular phenotype. So basically, I'll just take you through this here. You can see here, this is the % level of RNAi silencing of the RNAi SMART pool, giving about 80% knockdown. And the morphology of that cell shows an elongated phenotype in terms of nuclear displacement relative to control. And similarly, 3 out of the 4 siRNAs that resulted in knockdown, giving about 80% knockdown, also increased the nuclear displacement parameter. However, the siRNA number 4 targeting RNAi, which gave the least effective knockdown, produced the least effective phenotype in terms of nuclear displacement. So there's a clear correlation between the degree of silencing of RNAi and the phenotype. So I haven't shown you today some live cell imaging experiments that we did when we knocked down RNAi, but using the in-cell analyzer we could clearly show that once T cells were depleted of RNAi using the Accell siRNAs, that these cells failed to migrate, and we could clearly see that the tails of these cells were stuck on the substrate, on the anti-LFA1 substrate. And while the leading edge was capable of moving forward, the tail of the cell was stuck to the plate. So we wanted to see how does this translate in terms of T-cell migration. So we tracked the speed of the cells as they were migrating over the anti-LFA1 substrate, and this is quantified here in this next slide. And it shows that knocking down RNAi expression with the Accell siRNA decreases the T-cell speed on anti-LFA1. In parallel experiments, we also looked at the ability of T cells to migrate through a porous filter coated with ICAM1 towards the chemokine in the bottom of the transwell. And using T cell donors from 4 independent experiments, we were clearly able to show that knockdown of RNAi using the SMART pool siRNA reagent decreased the ability of the cells to migrate through a filter, thereby showing that RNAi is required for T cell migration in vitro. Now, up to this point, we had knocked down RNAi and placed the cells on an anti-LFA1 antibody. So we also wanted to check, well, would you see a similar phenotype if we plate the cells on the physiological ligand ICAM1? So we did this experiment. So the cells which have RNAi knocked down are in black bars, whereas the control siRNA T cells are the white bars. When we placed the cells on anti-LFA1 as expected, these cells elongate. And when we also place the cells on the physiological ligand ICAM1, these cells elongate as well. In parallel, we wanted to see what was the polarization of T cells like on other integrin ligands, for example, integrin ligands that are specific for beta-1 integrins. And similar to migration on the LFA1 ligand ICAM1, we found that migration of T cells on both VCAM1 and fibronectin was increased once RhoA was knocked down. Therefore, RhoA is required for migration on multiple T cell ligands. So going back to my first slide, I told you that T cells are important for defense against tumors and pathogens. They're involved in inflammatory disease and autoimmune disease, and they may lead to T cell leukemias and lymphomas, and they're also the site of infection by HIV. So I think where the Accell RNAi screening technology will have great applications will be in all of these types of assays. For example, some RNAi screens have been done in T cells to date, but they've primarily used GERCAT T cells, the transformed T cell line. So I think being able to perform a screen in a primary human T cell will really help to drive this field forward. So in summary, we've demonstrated the feasibility and applicability of Accell siRNA library screening in primary human T cells. We show that there's no specialist equipment required, such as multi-well plate electroporators, or you don't need any expert knowledge in viral vectors for the delivery of these siRNAs into primary human T cells. We show that the Accell siRNAs are efficiently delivered into the cells, result in robust silencing of gene expression, and importantly, they're available in library format for RNAi screening studies. And from our screening study, using the library targeting 72 distinct genes, we identified RhoA from this library screen as a crucial regulator of T cell polarity and migration. We also showed that multiple assays can potentially be performed from a single RNAi library screen, and this increases the comprehensive biological readouts that can be performed from a single screen. And of course, these screens or these assays can be coupled with the Accell library and other plate-based assays, for example, ELISA assays to measure, for example, in terms of T cell cytokine production, transwell assays to monitor T cell migration. As we have shown here, an imaging assay using an image-based high-content analysis platform, while they can also be coupled with various gene expression reporter assays using fluorescent or luminescence-based methods, or other signaling assays such as calcium flux. So with that, I'd just like to thank everyone for listening today. I'd like to thank everybody who's involved in this work from both Trinity College and from Dharmacon. So thank you very much.
Thank you for those interesting and informative presentations. While we are getting ready for the Q&A session, 2 polling questions will be appearing on your screen, and we would appreciate your answers to these questions so that we may follow up appropriately with you. Thank you. During the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question from our audience: is there any information on efficiency of Accell siRNA in activated T cells?
Could you repeat that question again there, please?
Certainly. Is there any information on the efficiency of Accell siRNA in activated T cells?
Yeah, well, the data I showed today was all activated T cells. So we were able to show 100% delivery of the siRNA into the T cells. In terms of non-activated T cells, there is a lot of data out there as well in terms of the use of Accell siRNAs for these cell types in terms of knockdown, efficiency, and also delivery. And we have reviewed some of these studies in our biochemical journal publication in 2013 for anyone who is interested.
Thank you. Our next question, 96 hours of incubation. Is there a need to change the medium once?
I'll go ahead and take that one. So I did see in the Q&A there was a lot of questions about incubation times and the serum requirements. So I'll try to address a lot of them in this response. So the first thing I want to point out is that the reason that we require serum-free or low serum conditions, it's not a stability problem with the siRNA. The problem is that the BSA in serum will bind up the cholesterol on the Accell siRNA and really prevent it from getting into the cell. So we really need to reduce that during the time that Accell is working on getting in, if that makes sense. So when we say low serum, our own testing showed that serum up to about 2.5 to maybe 3% was still not interfering with knockdown enough, but did boost the cell viability in cells that were a little more sensitive to those low serum conditions. That's going to be very cell type dependent, but the recommendations you'll see in our protocol will say 2.5 or just less than 3%. We have shown, and many publications have shown, that adding serum-containing media back at 48 hours. So if you have a need for a media change anyway, you can use serum-containing media, because by that 48-hour time point, we are able to know that the siRNA is getting in. But we just need to wait. We typically do recommend, again, though, that 72 hours or more before you actually look at knockdown. So hopefully that answered. Again, so much of this is just, as you would do with any new experiment, you have to optimize. You have to make sure that your cell density, your cell type is balanced out with the type of results you're getting from the Accell knockdown.
Great. Our next question, have you done Accell siRNA in the knee or cartilage of the mice?
Oh, that is a great question. We ourselves have not done that. I am almost positive that another group did do a delivery to, I want to say it was to the gums of rat, to actually do a periodontal model. We do have over 70 references available online that are kind of sorted and characterized by cell type or in vivo cell model. That's a great reference. If the one I'm thinking of is not on the list, I will get back to that person specifically with the reference I'm thinking of.
Our next question, what can be the highest concentration of serum in the Accell delivery media?
Oh, good question. I may have answered that with my earlier response. Again, you can add as much serum as you might need to the Accell media, but we do recommend 3% or less to avoid interfering with the Accell delivery itself.
Our next question, how did the T cells respond to the serum-free conditions for Accell delivery? Were the conditions difficult to optimize?
No, the conditions weren't difficult to optimize at all. We just resuspended the cells in the Accell delivery medium. Now, we were using activated T cells. So when we routinely culture activated T cells, we add interleukin-2 in the medium. So we added interleukin-2 in the medium during the knockdown period. So we saw no loss of cell viability during this period.
Thank you. Our next question, are you planning any additional screens?
Yeah, I'll take that one again. We hope to be in the future, depending on funding, we would like to do some larger screens with Accell libraries. So yes, for sure.
Great, thank you. Another question, can I use Accell with electroporation or lipofectamine to improve transfection efficiency?
That's a good question. I think that one came in early on, maybe before we got into some of the details. So Accell siRNA, of course, could be used with a transfection reagent or with electroporation or some other instrument-based technology. But that's sort of like putting your car in the back of a semi to drive it. It's really not necessary. You're not going to get improved results from either of those other delivery technologies because of the Accell. Accell really works on its own, kind of as naked RNA getting into the cell itself. So you could use it, but there's no reason to not just use conventional unmodified siRNA for transfection reagents or electroporation use.
Great. Thank you. And we have time for one last question. We have it here. Is it sometimes required to add Accell repeatedly during the experiment duration for an optimal K-down?
Oh, that's a great question. One of the things that we tested early on that I did not really have time to work into the presentation was that we were able to get extended duration knockdown by repeatedly treating the cells with Accell. So it was sort of an every other media change. We added back Accell, and we were able to get knockdown out to 20 or even 30 days. There's a publication on, that's in our references where they got 10-day duration knockdown in primary neurons. So it's not necessarily going to give you stronger knockdown by repeatedly dosing, but because it is so gentle to the cells, you are able to get a longer duration of silencing by repeatedly adding it back to the media.
Thanks so much. That's all the time we have for questions. I'd just like to let our audience know if you submitted a question through the Q&A button and it was not answered, someone will get back to you regarding your inquiry. If you have further questions that you did not submit during this broadcast, please email them to the Dharmacon team at ts.dharmacon@ge.com, and someone will be happy to assist you. I would like to thank our speakers, Louise Baskin and Michael Freeley, as well as our sponsor, Dharmacon, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through November 12, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. And we invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you very much. See you next time. Goodbye.